Introduction to Modern Bioinformatics Research. Bioinformatics has rapidly evolved into one of the most critical fields in biological research, driven by advances in high-throughput technologies, computational power, and large-scale biological data generation. From analyzing vast genomic datasets to modeling complex biological systems, bioinformatics plays a central role in modern research, providing insights into disease mechanisms, evolutionary biology, drug development, and more. This section delves into the latest trends and breakthroughs in bioinformatics research, focusing on areas such as single-cell genomics, artificial intelligence, AI, computational drug discovery, microbiome research, and more. 1. Single-cell genomics, unraveling cellular heterogeneity background. One of the most exciting areas of bioinformatics research today is single-cell genomics. Traditional genomic analysis methods rely on bulk sequencing, which averages the genetic information across millions of cells. While this approach has been essential for many discoveries, it masks the heterogeneity between individual cells. Single-cell genomics allows scientists to investigate the gene expression patterns, mutations, and epigenetic modifications within individual cells, providing unprecedented insights into cellular diversity. Current advances. With the help of bioinformatics, researchers have developed methods to analyze single-cell RNA sequencing, scRNA-seq data, which allows them to study gene expression at the resolution of a single cell. Key bioinformatics tools and techniques include cell clustering algorithms, techniques like TSNE, T-distributed stochastic neighbor embedding, and UMAP, uniform manifold approximation and projection, allow for the visualization of single-cell data, grouping cells with similar gene expression profiles. These clusters can reveal distinct cell types and subpopulations. Pseudotime analysis. Tools like Monocle can infer the developmental trajectories of cells, mapping out how stem cells differentiate into various cell types over time. Applications and case studies. Cancer research. Single-cell genomics is revolutionizing cancer research by uncovering the cellular heterogeneity within tumors. For example, scRNA-seq has been used to identify rare cancer stem cells, which are thought to drive tumor growth and resistance to therapy. Immunology. Single-cell technologies are also being applied in immunology to study the diversity of immune cell populations, particularly in the context of infections, autoimmune diseases, and vaccine responses. 2. Artificial intelligence and machine learning in bioinformatics. Artificial intelligence, AI, and machine learning, ML, have become indispensable in bioinformatics, where they are applied to analyze and interpret large, complex datasets. From predicting protein structures to identifying disease biomarkers, AI is reshaping the field by enabling faster, more accurate analyses that would be impossible with traditional methods. Key AI techniques in bioinformatics. Deep learning models, particularly convolutional neural networks, CNNs, and recurrent neural networks, RNNs, are used to analyze sequences, images, and even protein structures. For instance, AlphaFold, developed by DeepMind, revolutionized protein folding predictions by using deep learning techniques. Natural language processing, NLP. NLP tools are used in genomics to extract relevant information from vast repositories of biological literature. Tools like BERT, bidirectional encoder representations from transformers, are employed to help mine text data for research insights. AI in drug discovery. Machine learning algorithms are increasingly being used in computational drug discovery to predict how molecules will interact with proteins. This approach accelerates the drug development process by screening billions of compounds in silico, identifying potential drug candidates before clinical testing. Applications and case studies. Protein folding with AlphaFold. AlphaFold made a breakthrough in predicting 3D structures of proteins from their amino acid sequences. This tool provides crucial information for understanding protein function and has far-reaching implications for drug design and disease research. AI for personalized medicine. Machine learning models are being developed to predict patient-specific responses to drugs based on genomic and clinical data, paving the way for personalized treatment regimens in cancer and other diseases. 3. Microbiome research. Analyzing complex microbial communities. The human microbiome, comprising trillions of microorganisms living in and on our bodies, is a key research area in bioinformatics. Recent advances in sequencing technologies, coupled with sophisticated bioinformatics tools, have enabled researchers to study the composition and functions of microbial communities at unprecedented depth. This has led to new insights into the role of the microbiome in health and disease. Key tools and techniques. Metagenomics. Metagenomics involves sequencing the genetic material of entire microbial communities, bypassing the need for culturing individual organisms. Bioinformatics tools like MetaPHLEN, metagenomic phylogenetic analysis, and Kraken are widely used to classify microbial species and their abundance in various environments. Functional profiling. Bioinformatics tools like HUMANN, the HMP Unified Metabolic Analysis Network, are employed to study the functional potential of microbial communities, allowing researchers to predict which metabolic pathways are active in different environments. Applications and case studies. Gut microbiome and health. Bioinformatics has played a critical role in analyzing the gut microbiome, revealing its influence on digestion, immune function, and even mental health. For example, dysbiosis, imbalanced microbiome, has been linked to conditions like inflammatory bowel disease, IBD, obesity, and depression. Environmental microbiomes. Metagenomics is also applied to study microbial communities in soil, oceans, and other ecosystems. Researchers use bioinformatics to monitor how microbial communities respond to environmental changes, such as pollution or climate change. Fourth, CRISPR and genome editing. CRISPR, clustered regularly interspaced short palindromic repeats. Technology has revolutionized genome editing, providing a powerful tool for precisely modifying DNA in living organisms. Bioinformatics plays a critical role in CRISPR research, from designing guide RNAs, gRNAs, to analyzing the off-target effects of gene edits. Key tools and techniques. CRISPR gRNA design. Bioinformatics tools like CRISPR-Cas9 are used to design gRNAs, which guide the Cas9 enzyme to the target DNA sequence for editing. Tools such as ChopChop and CRISPR help researchers select optimal gRNA sequences while minimizing off-target effects. Off-target analysis. One of the main concerns with CRISPR technology is the potential for unintended edits, off-target effects. Bioinformatics tools analyze the genome for regions that closely resemble the target sequence, predicting the likelihood of off-target cuts. Applications and case studies. Disease treatment. CRISPR is being explored as a potential treatment for genetic diseases like sickle cell anemia, muscular dystrophy, and cystic fibrosis. Bioinformatics tools help researchers evaluate the safety and efficacy of CRISPR-based therapies by analyzing genomic data from treated patients. Agricultural genomics. CRISPR is also being applied in agriculture to develop crops with improved traits such as drought resistance, increased yield, or enhanced nutritional content. Bioinformatics helps identify the genes responsible for these traits and evaluates the impact of genetic modifications. 5. Epigenomics and transcriptomics. Epigenomics and transcriptomics are emerging fields that focus on gene regulation and expression. Epigenomics examines chemical modifications to DNA and histones that affect gene activity without changing the DNA sequence itself, while transcriptomics studies the complete set of RNA transcripts produced by a genome. Key tools and techniques. DNA methylation analysis. Bioinformatics tools like Bismarck are used to analyze DNA methylation patterns, which play a crucial role in regulating gene expression. Changes in DNA methylation have been linked to cancer and other diseases. RNA-seq analysis. Transcriptomics relies heavily on RNA sequencing, RNA-seq, a powerful technique for quantifying gene expression. Bioinformatics tools like DSEC2 and EDGE-R are widely used to identify differentially expressed genes across various conditions or time points. Applications and case studies. Cancer epigenomics. Epigenomic studies have revealed that changes in DNA methylation and histone modifications are key drivers of cancer development. Bioinformatics tools are essential for analyzing these epigenetic changes and identifying potential biomarkers for early detection or targeted therapies. Transcriptomics in disease research. Transcriptomic studies have provided insights into gene expression changes associated with diseases like Alzheimer's, Parkinson's, and diabetes. These studies help identify potential therapeutic targets and improve our understanding of disease mechanisms. Conclusion: bioinformatics research is at the forefront of modern biology, enabling breakthroughs in areas such as single-cell genomics, AI-driven drug discovery, microbiome research, CRISPR genome editing, and epigenomics. As computational power continues to grow and new technologies emerge, bioinformatics will play an even more critical role in advancing our understanding of biology and improving human health.